Well, thanks, Jim. Actually, the first guy he showed and I both have mustaches except for me, and otherwise I don't think I have much in common with any of these other people. It's appropriate that this meeting is in Chicago. I'm getting over one of the worst colds I've ever had, and you may all recall this is like the 1994 NBA Finals when Michael Jordan came out with a fever of 102 and still, won the game. So hopefully I'll get through this talk without paroxysms of coughing. Anyway, that was a terrific introduction because it really led into a lot of the stuff that we've been trying to do both within our own center and then within SWOG in terms of not only discovery and pushing the envelope of the science, but also how can we apply these things in clinical studies and clinical practice. I want to start out, first of all, though, and I have some very real conflicts of interest here. I've received laboratory and clinical research funding from Veridex for a long time now, over 15 years, and they're the ones who make cell search. I have 2 provisional patents pending with them regarding CTC clinical utility and another one on my own independent of that regarding novel technology. But I don't have any financial interest in Veridex or J&J. I have a lot of intellectual conflicts of interest, I guess. I've been pretty committed to them. And a lot of this talk will involve their work, although I don't believe it's the end game. I think there's a lot more to do, as Jim said. Some of you have heard me give this, show this slide before, but I think all of us who are getting involved in tumor biomarker work need to fully understand these 3 semantic areas. These were first articulated by Steve Teutsch in the Evaluation of Genomic Applications in Practice and Prevention Initiative, so-called EGAP initiative, published about 3 years ago, related to genetic testing, but they apply to, frankly, any diagnostic testing. And that there are 3 critical components to actually getting an assay into clinical use. One is analytical validity. Is this really an accurate, reproducible, and reliable assay? And that's a real concern, especially with circulating tumor cells. These assays are not easy to do, and there is a lot of variability, even with the highly validated ones. The second term is clinical, or what the IOM committee added to called biological validity. And that is, does the assay actually identify a biological difference within the group you started? So does it split the group of patients into 2 groups that either are biologically different or clinically different? If it doesn't do that, it's not worth pursuing any further. But that doesn't mean it's clinically useful. It doesn't mean that those of us who wear white coats and take care of patients would use that assay just because the p-value is 0.04. It just means at least you're on the right track. And what we really want is clinical utility, and that is, do the results of the assay lead to a clinical decision that's been shown with a high level of evidence to improve outcomes compared to not knowing the assay at all. And that's a high bar, but I think we need to really use that bar in terms of introducing these things into the clinic. That bar has not been used in the past for diagnostics. It's a long story regarding the regulatory issues in the United States and how we look at diagnostics and devices as opposed to drugs. And many of you have heard me say and pound the table that I think we need to consider tumor biomarkers the same way we develop drugs, and that a bad tumor biomarker is every bit as bad as a bad drug, and we need to value them the way we do drugs. Now, there are 2 or 3 issues about circulating tumor cells. One of those is to enrich them from the sea in which they find themselves, the millions of white cells and billions of red blood cells and platelets within a 10 cc tube of blood, for example, and then to characterize them and make sure you aren't actually measuring red cells or white cells or normal epithelial cells or benign from malignant cancer cells. And these are all huge challenges. The first representation of a circulating tumor cell that I know of was in 1857 by a pathologist named Ashworth, who during a post-mortem just did a smear of blood from the deceased patient and found cells and drew pictures of them. I'm not going to show you the picture, but they absolutely look like cancer cells. But it's taken 150 years or so to actually translate that into a usable assay that we can utilize in the clinic. There have been a number of techniques and assays to detect them. I'm not going to spend a lot of time on this, but it's separating cancer cells from normal cells or epithelial cells from hematopoietic cells based on differences in size or motility or expression of a variety of biological characteristics. And this is a slide that Richard Cody made. I'm going to talk about some of his work in just a minute later within SWOG regarding 4 or 5 different ways to do these things. And there are many, many other ways to do these things. Actually, is Stephanie here yet? I don't see her yet, but I didn't put MagSweep on here. I could have. And in fact, the next 3 slides are just almost the same slide that Jim showed you of different ways to capture and evaluate circulating tumor cells. And I'm not going to spend time on this, just to show you that there are lots of people involved in this right now. Of all these, as Jim already said, CellSearch is the only commercially available assay approved by FDA for routine clinical use. It has enormous analytical validity. I'm pretty confident in its reproducibility and accuracy. And the routine clinical use that has been demonstrated for it is to monitor patients with metastatic breast cancer, prostate cancer, or colorectal cancers. This is the closest thing to a commercial that I will show you, and I only show it to you to give you an idea of the amount of work and the millions of dollars of R&D it's taken to actually get this assay into the clinic. It's basically now, Jim just called it a big box. I like that. It's really a black box assay. It's kind of hard to screw up, frankly. Even I can do it, although my technicians and postdocs won't let me in the lab to do it. But when I sneak in at night, I can actually run this assay. One thing we found was, for example, when we first began to utilize the techniques, the company was actually called Immunicon. They've now gone out of business and sold out to Veridex. But we were collecting samples in our clinic at Georgetown. We were collecting 2 samples. One we were running in my lab at Georgetown. The other we were sending off to Immunicon in Philadelphia, and we were getting much better results than they were. I assumed it was because I had this really super-duper technician, Solomon Kabidi from Ethiopia, who was working hard and staying all night, and they had some lazy American technician who they couldn't get to work at all. Turned out that wasn't the answer at all. The reason we were getting better results was not because Simon was a great tech, or Solomon was a great tech, he was, but because the cells die very rapidly once you get them out of the human body. In fact, they are dying inside the blood. We've now got data to tell us that. And that if you don't assay these cells within the first couple of hours out of the body, they're gone, unless you put them into a tube that has a fixative. And so the company very quickly put together these cell-safe tubes that have a certain amount of fixative. It's a proprietary secret. I don't know what it is, but if you smell it, there's formalin in that tube. I don't know what else is in that tube. In addition, they've worked out the magnetics and the physics and the cell tracks, and so it's a very nice assay. This is the meat of the assay. At the end of the day, you get a printout. What you've done is capture the cells immunomagnetically with an antibody against EpCAM, and then you come back in with immunofluorescently labeled antibodies against cytokeratin. Actually, first you come back with DAPI, which tells you that the event that you're identifying is in fact nucleated. And then cytokeratin, which tells you that this is an epithelial cell, and epithelial cells don't belong in the blood. So presumably in a patient who has cancer, it's almost certainly a cancer cell. In fact, I'm not going to show you the data, but they're pretty good data to demonstrate that. For screening, that may not be true. All of us have an occasional normal epithelial cell floating around our blood, so it's not clear that these assays are going to be good for screening. And then a third antibody, or second antibody against CD45, and that's to show that whatever you've detected is not a white cell. No matter how careful you are, there are white cells that come across in the assay, partly because the white cells are actually trying to eat the cancer cells, so you can't separate them altogether. And then the machine puts together a composite image, and the only thing actually that's not automated now is a human being has to go through and read these and say, this is nucleated, cytokeratin positive, and CD45 negative, and that gets a check mark, and then the machine adds up the check marks and tells you how many CTCs you have. I want to point out there's a fourth channel here. This was initially developed for standardization between runs. It turns out the assay is so well standardized between runs, we don't need to do that anymore. And that's left us with a fourth channel in which we can phenotype these cells as long as we can develop an antibody that detects an antigen that is expressed in sufficient abundance that it doesn't get wiped out by the cytokeratin and that survives fixation and doesn't float out of the cell when you permeabilize it. Those are not trivial issues, by the way. It's taken us about a year to work up each of the antibodies we've done, but it can be done. I'll show you some data in just a minute. All right, for sure this assay has clinical validity, and the question is, what is the use you would use it for? Screening right now, I think, is off the table, frankly. Prognosis in early stage disease is getting increasingly interesting. I've not done any of that work, but I'll show you some of that in a minute. Monitoring metastatic disease is where I've lived my life up to now, and molecular characterization of cells once we identify them. So this is a model that all of us are oncologists live in terms of solid tumors, and you go from relatively normal ducts out to ducts that are chock full of cells that pathologists would call cancer that are invading out into through the basement membrane and ultimately metastasizing either as micrometastasis or ultimately macrometastasis and death of the patient. And so, of course, clinically we have premalignant conditions, and then these are the kinds of therapies that we apply to these places. What we're interested in is, can we detect these cells either in patients who already have metastasis or in the primary? And these are the data that really got us where we are. This is Massimo Cristofanelli was the first author in this paper we published in the New England Journal about 10 years ago now. And this was a prospectively done study of 175 or so patients in which we took the first 100 patients and worked out cutoffs and determined that indeed we could detect a group of patients with elevated cells, that being more than 5 CTCs per 7.5 mils of whole blood, and that they have a worse prognosis if their cells are elevated prior to initiating some sort of therapy for metastatic disease, compared to those who don't have elevated cells. Now, they all have metastatic disease, so they're all continuing to relapse over time, but clearly this was a prognostic assay. And then with no monkey business, we applied it to the second set of patients who were enrolled in that trial and got almost identical results. In fact, I tell my postdocs, if you're going to cheat, this is how you want to cheat. You don't want to have identical results. That's too obvious. You want to have results that look pretty good but not great, so this looked pretty good. There's no cheating involved here, by the way. Since this is not a breast cancer assay, it's actually an epithelial cell assay, it makes sense that it would work in other epithelial cancers, and indeed this slide shows you that CTCs are elevated in many of the epithelial cancers, and now there are data that look almost identical to our data in colorectal, and also from Johan de Bono, and Howard will be talking more, I'm sure, about prostate cancer, and I'm going to show you some data from SWOG. So this assay looks pretty good. The real issue, though, to me was not whether a pre-treatment elevated cell splits the curves into two. That's clinical validity, but the clinical utility would be, so what? What can I do about that? And the real issue for me was whether or not, if very quickly after starting a new therapy for metastatic disease, we could identify a group of patients who didn't seem to be responding, and we could switch therapy right away instead of waiting the usual 3 to 4 months to see if they are responding or not. And so these are the data from the same trial I just showed you, but this is first follow-up rather than baseline. And what you can see here is the green line, the patients who don't have elevated cells at first follow-up, again, continue to do pretty well. The red line now has shifted to the left because about 30% of the patients on the red line at baseline have responded, have lost their cells, are now on the green line, and now the red line is even steeper because these are patients who don't seem to be responding to their treatment. Still, though, in my opinion, that's clinical validity but not clinical utility. And so the question is, although many of these patients relapse or progress very, very rapidly, some do not, and we also don't know if changing therapy right away would make a difference for those who do progress rapidly. And so within Southwest Oncology Group, led by my colleague Jeff Smiraj from the University of Michigan, we started SWOG SO500. We focused this on patients who had first-line chemotherapy in the metastatic setting, and that's because that's a homogeneous group of patients for whom we thought it was most likely to be a successful study. If at baseline they didn't have elevated cells, then they were treated with whatever their physician felt they should be treated with, and they're on that green line. But if they had elevated cells at baseline and then received one cycle of chemotherapy, whatever their physician felt they should receive, if they cleared their cells, they're on that green line, they stayed on whatever their doctor started them on. But if they didn't clear their cells, they're on that red line, and they were randomly assigned to either continuing until they had classic evidence of progression by clinical or radiographic means, which is standard of care, or switched right away to whatever other chemotherapy their doctors thought would be appropriate for them. These are the numbers of accrual. We screened 550 patients, and ultimately we've randomized 123. We got just what we thought, which is that half of them didn't have cells at baseline, half of them did have cells at baseline. That's been seen over and over and over again in terms of the cutoffs. And this study was closed to accrual March 15, 2012, fully accrued. I thank all of you who participated in this. And now we're waiting to see if we'll meet our primary endpoint, which is does it improve survival by switching therapy right away as opposed to continue to treat a patient with an apparently futile therapy for several months. The second issue is whether or not these could be used for prognosis in early stage disease. This is a graph from the SUCCESS trial run in Germany, and data almost identical to this were just published out of MD Anderson by Luki and colleagues in The Lancet within the last few months. And they show that patients who have one cell out of about 22 mils, so a lower cutoff and a larger volume, do have a worse prognosis than those patients who didn't. Now, again, in my opinion right now, this has clinical validity but not clinical utility. I'm not sure what to do about that. It is not clearly independent of the other prognostic factors we already know about, but at least it is of interest and suggests to us that we can begin to look at CTCs in early disease and perhaps do interesting things with those. So what are the major hurdles for us? One is sensitivity. Jim's already alluded to this. Only 50% of patients have more than 5 CTCs in the metastatic setting, and in the adjuvant setting it's even less than that. And that may be because we're only pulling out cells that make EpCAM, and it's very possible that these cells down-regulate EpCAM when they go off to go somewhere else. It may be because we can only interrogate 10 to 40 CCs at most. The IRBs get pretty reluctant to allow us to exsanguinate patients to look at all their blood. It's because we have to manipulate the cells as we're doing the assays, and you always lose some. So there are real issues with sensitivity, but as Jim also said, the real concern I have is many investigators are improving sensitivity and forgetting the fact that specificity is probably as important as sensitivity in a diagnostic assay. Not just specificity versus white cells, but specificity versus one kind of cancer cell versus another. And we've already shown, I'm not going to show you the data, but that about 30 to 50% of the cells in blood are already apoptotic. Those cells probably have no meaning and probably shouldn't be counted. It's the cells that aren't apoptotic that you want to count. And there are a variety of people beginning to address this, but I think everybody who's in the field needs to be very cognizant of the fact that just finding more cells is not a better assay. Okay, what we'd really like to do is characterize the cells, and as Jim pointed out, use this as what we would call a liquid biopsy or a real-time biopsy. Of course, what we love to do is biopsy the patient's cancer and biopsy every site of her cancer all the way through her course. That's not practical. And so the question is whether we could use markers on CTCs to do that. We and others have already now begun to report that we can do ER, HER2, BCL2, Ki67, apoptosis, IGFR1, AR, Momentum, and even some others now. So we're beginning to see things come with cell search and with the other assays as well. And I'm going to skip over that real quickly. And I want to just show you some data that we generated at the University of Michigan. Costanza Paletti, who's an extraordinarily talented postdoc in my lab, has done most of this work. We actually had proposed to place this into a new SWOG protocol. Unfortunately, the North American Breast Cancer Group Steering Committee has now turned down the protocol. George Somlo was trying to get through, but I still think there's room to move this in. And what we're doing is looking at patients with metastatic disease. Of course, when we have a patient with metastatic disease, I tell my patients the bad news is you have metastatic breast cancer. We're probably not going to cure you. The good news is you have metastatic breast cancer because there are so many options, and this slide is an example of how many options we have to reach a secondary objective, which is to keep you feeling as good as you can for as long as you can. If a patient is ER positive, we'll treat you with endocrine therapy until we believe it has quit working, and when we believe the patient is refractory to endocrine therapy, we go to chemotherapy. If a patient's ER negative, we go right to chemotherapy alone. Our hypothesis was that maybe we could identify a group of women with ER-positive breast cancer who won't benefit from the next endocrine therapy, and instead of giving them a futile therapy for several months, just as if they were ER negative, go right to chemotherapy. And in fact, every trial I've ever seen of endocrine therapy, whether it's first line, second line, or third line, has a drop-off in the first couple of months in the Kaplan-Meier curves, suggesting that there's a group of people who are ER positive but are intrinsically resistant to endocrine therapy and probably would be better off treated with chemotherapy. So we chose 4 markers, ER, BCL2, as markers of sensitivity, HER2 and Ki67 as markers of relative resistance. I won't go through why, but if you're a breast cancer maven, you know that these are big players in the 21 gene recurrence score. And we had antibodies for them, and we could work the antibodies into the cell search assay. This is just an example. These are MCF7 cells and SKBR3 cells. MCF7 cells are positive for ER and relatively negative for HER2. SKBR3s are positive for HER2 and relatively negative. And the last channel is that fourth channel I told you about. This is for anti-ER, and what you can see, whoops, didn't want to do that, is that the MCF7 cells are positive, and that's nuclear staining, just as we'd expect, and the SKBR3 cells are negative. So these are real patients. We've run a pilot study of about 50 patients now looking at ER, BCL2, HER2, and Ki67. This particular patient, for example, was very positive for ER and BCL2, very negative for HER2, and modestly positive for Ki67. And we've worked out what we call a CTC endocrine therapy index, and I won't bore you with how we do that. But low is good, high is bad, just like tissue grading. And so this patient had a low CTC-ETI. We would have predicted that she would be respond or benefit from endocrine therapy. And this patient had almost no ER, although her tissue, by the way, was ER positive. BCL2 was positive, HER2 was negative, and Ki67 was actually much higher. And this patient had a high CTC-ETI, and in fact, she was already refractory to endocrine therapy when she entered into this pilot study. So we've done the things I've told you about. There are a number of other technologies that are starting to look at other things, and I'm going to skip over that. And then finally talk briefly about some things that SWOG has been doing in prostate cancer. These are not my data. I want to give thanks to Nick Vogelsang, David Quinn, and Amir Goldkorn for sharing their slides. I don't believe any of them is in the room, but they graciously gave these to me. SWOG SO421 was a phase 3 study of docetaxel and atrasentan versus docetaxel and placebo. And to make a long story very short, those are the Kaplan-Meier curves. The drug didn't work, but they did CTCs in these patients. And as expected, and not different from Johan de Bono and Howard Shear's work, CTCs were highly prognostic at baseline in this group of patients. Maybe more importantly, they also did a second assay using Richard Cody's slot microfilter. What I just showed you was cell search. And in this case, they're isolating cells based on the difference in size and fluidity and not based on expression of EpCAM. And I won't go through the details except to say that you get these cells trapped on the microfilter and you can do a number of things with them. And Amir has done telomerase activity in these cells. And again, I'm not going to go through the assay completely because I'm not sure I understand it, except to say that then applying that to the same study, what they have found is that if you have cells and they are telomerase active, those patients had a worse prognosis than if you either didn't have cells or had cells that were telomerase not active. And I think this is pretty exciting. It really begins to suggest we can break these cells into their biological characteristics and begin to take advantage of that. And this is just from Nick. He asked me to please thank the co-authors and the other people who are involved in this work. So to make a long story short, I think the world is our oyster. I think there's a lot going on. I'm excited to hear what the other speakers have to say. These are the people in my lab who have done the work. This is Costanza, and she is from Italy, and believe it or not, that is a cancer cell. That's a circulating tumor cell. She's looked at hundreds of thousands of these cells and found one that was shaped just like Italy. And so I thank you very much. This is Breast Cancer Awareness Month. I'm sure you're all aware of this. And I will also say this is an MCF7 cell, believe it or not. The only photoshopping here is we just changed the colors and made DAPI green and made the cytokeratin pink. And so thank you very much.